So now, Dr. Soltesz, if you can jump on, and then we'll come back to all of these and come back to you three also as we're having further discussions. We're just going to start with the overview. All right, thank you, Ivan.
Thank you, Eric, and thank you, Tracy. So I also don't have any slides. Tracy asked me to, quote, speak eloquently, which I guess she didn't mean to say, be blabby, and she made sure to restrict the comment to 10 minutes. So I shall try to be very eloquent, but short. So a few things about what I heard, and as a basic scientist, I just reflect on what I heard, and I hope actually that some of you will disagree with me, because otherwise there is no point of having a meeting, right? So, first of all, what I heard repeatedly is the puzzle that there are multiple causes of LGS, yet similar syndromes. So that's number one. I like to comment on that a little bit from my perspective. Also about the nature of the EEG, which is this abnormal EEG, the spike in waves and the fast activity. And finally, the developmental angle, which is, I think, is obviously for LGS an important component, and particularly from the epileptic network perspective, what we heard from Dr. Peters, the developmental expansion of the epileptic network. So from my perspective, and it applies to the epilepsy field in general, but also and maybe particularly to the LGS syndrome field, what I would call the macro-micro disconnect. And I apologize to those of you who have heard this comment before, but I think from my perspective, again, it's a critical one, which is that the signatures of a disease that we look at, whether it's behavior, whether it's EEG, or whether it's fMRI, they are fundamentally macroscopic measures, but the brain operates at the micro level, at the level of ion channels, synapses, and cells. So undoubtedly, the macroscopic descriptors are crucial and very important, both for the clinic, for obvious reasons, but also even for people who cross between the clinic and the basic science, like we heard from Jennifer Jalinas making the comment, I believe it was today, that the nature of that slowing of the background and the disruption of the normal activity, how that may relate to the cognitive deficits. So these macroscopic measures are important, but what I personally would like to get at, before I die, is the Hubble telescope for the brain, right? Particularly wouldn't that be wonderful, where you could see every single cell being active in a child's brain, right? So we don't just record the macroscopic activity, which is essentially a smeared out summation, even if you use a local electrode, but that we would be able to really look at, like with the Hubble telescope, a huge constellation of neurons in any part of the brain. Now, I have to say that there are enormous advances towards this. And let me just reflect on that from the basic science side. So, when I was a PhD student, I went to study in a lab that was able to record from a single neuron, and that was a big deal from an in vivo animal. So that was a huge deal. Whatever, 30 years later, now we can do routinely thousands of cells, and we can follow the same cells over time. So we can really look at that. Now, of course, 1,000 of cells is not, 1,000, 2,000, 5,000 cells is still a very small minority of the total brain that one would like to see with the, imaginary Hubble telescope. But we are getting there. In addition, we are not just able to look at the spikes of neurons, which are important, but fundamentally they are just the outputs, right? We are able to now look at for example, with calcium, but especially with the enormous advances made with voltage-sensitive dye imaging in vivo, where you can look at multiple cell types at the same time and look at the subthreshold activity as well in vivo. And I just want to say that there are very interesting technologies where you can, for example, look at like Mark Schnitzer developed the skull technique, where you can actually replace the skull with a curved glass, and then you can put your 2-photon microscope at multiple places and look at how the activity, what is the activity dynamic like at a single cell resolution. So I think that these kind of advances are going to be critical for epilepsy in general, but also for LGS in particular. In addition, manipulation of the circuits are becoming better and better. And I just mentioned a few things in really fast. So for example, wouldn't it be lovely to know that what happens when a seizure starts exactly? One way to do that would be, and we have a tool like that now, where you would record the activity and then flashlight and somehow label only those cells that were active at that point. In 10, 20 years ago, people did CFOS for this, but now we can do the flash of light that would label only cells that were active at that particular time. There are tools for this now. They're not perfect, but the tools are getting better and better every time. Another huge advance that I think eventually will make it to the clinic is really the manipulation of cells with light, or by some other means. And a huge step forward was when techniques were developed to actually being able to flash the light from the outside of the skull, so you didn't need to put an optrode inside and manipulate the neurons optogenetically that way. That was huge. Another one that was huge is the ability of inserting particular proteins in particular neurons using viral vectors without having to again enter the skull and inject the brain. So this is now real. So for example, retroorbital injections of AAV vectors can be designed in such a way that these vectors then spread to specific cell types in the brain and, for example, express whatever protein you want in those cells. And we all know that these kind of approaches have enormous power. The mRNA vaccine was like that essentially, right? And many of us benefited from that. So I think that although it's not right now a way to do it, but AAVs are being used in humans as we speak, and it's an accelerating field. So I think it's a very exciting one that should be, in my humble opinion, pushed to the LGS field as well, increasingly. And in order to move on, because I don't want to speak too long, just a few more minutes, I just want to say that I think that the field is enormously benefiting from these exciting new models. Because for people like me, I always kept an eye on LGS, but I never entered the field, mostly because of the lack of good models. But I think it's very exciting that new organoid, fish, and even mouse models are now coming online that are increasingly enable researchers to really study the full brain, or at least a part of it, and really follow these networks, and the cellular level analysis of this network is now possible. That's really the key. Now, I would say that just from a theoretical point of view, the fact that we can now, for example, in a zebrafish, image all the cells in the brain, and a good chunk of them we can capture and in every brain area at the same time. Right now, the temporal resolution is still not perfect, but it's getting better. It's about half a second or maybe 200 milliseconds. Is huge in that regard, because now you have really, a situation where you can apply analytical tools and really study these networks as the seizures occur over time, and really chunk it and look in time and in space and analyze this network much better than that was possible even 5 years before. And the BRAIN Initiative in many ways really deserves a huge part of the credit for this. Finally, I just want to say a few words about the other aspects that I heard, which was for example, the cognitive deficits. So, the cognitive deficits field is, is something that, I have been interested in. And I just want to say that I was personally inspired by results in my lab that I didn't think would happen. And it's other people have had similar results, which is that even in a brain that, that has chronic epilepsy in a mouse, for example, it is possible to intervene and, while not completely reverse the cognitive comorbidities, but at least ameliorate them, that they get much closer to the control situation than before. That's really inspiring. So, for example, if we stop the seizures, make them shorter with optogenetic, closed-loop optogenetic intervention in a mouse model of chronic temporal epilepsy, and we intervene for 2 weeks and then test for memory, these animals, and importantly, during the test, we don't intervene. So that's a network that where we basically save the network, we decrease the seizure load for 2 weeks and then test for memory. And those animals clearly are much closer to control, to wild types, than those animals where we didn't intervene. So that to me is a very good sign, because it means that even though the cell loss, the sprouting, the ion channel expression changes, and all of those things occurred in these mice, but it was possible to phenomenologically at least, the comorbidities could be reversed. I also want to say that in terms of enormous advances in neuroscience that are occurring, that again should be applied to LGS, I want to highlight the issue of sensors for various neurotransmitters and neuromodulators. So, sensors for things like acetylcholine. In the past, you had to chew up the brain and then biochemically look at it, or you did some kind of a probe, which had a very kind of, it caused damage, and then you sampled, and the sampling rate was not very good. But now we have sensors, genetically expressible sensors, where in vivo you can look at the time scale of the actual neuronal dynamics, how the various neuromodulators change around the particular cell or at a particular synaptic compartment and so on. So, for example, for cannabinoids or many of the other stuff. And why is this important? It's because we heard repeatedly, for example, that, so LGS has this, difference between the waking state and the sleep state, right? The frequencies of seizures and all that can be different. Now, why is that, right? In order to get at that, biologically speaking, it must be the reason that it's neuromodulators that are at play there. And I would advocate for using these today available superb tools to get at these questions. And finally, on the point of sleep, it might not seem like a big deal, but one of the reasons I believe why the field of sleep research in epilepsy has been lagging behind was simply because it was very hard to study, for example, with imaging methods, epilepsy in sleep. It's now possible. So one, again, seemingly small change, but I think it's huge, which is that now we can study sleep in a head-fixed animal. And which what it really means is that you can apply very high resolution, cellular level resolution, very large scale imaging techniques or electrophysiology to really look at these circuits, how they behave in a control animal and in an epileptic animal at various stages of sleep, and really simultaneously look at the neuronal activity and the aforementioned neuromodulator dynamics. And I could go on, but I won't. I just want to say that, that the development is another angle where there are enormous advances, very exciting new technologies are coming online. Just one word on that, which is that, for example, many people, including, for example, Jennifer and Jelinas and Kathy Chavon and many others are thinking about how the normal activity gets transformed into an epileptic activity. And there are many researchers that are really looking at this problem from a developmental point of view, so that the networks that are being set up during development, how they determine the normal patterns of activity, so which groups of cells fire together, may be determined by when they were born early in development, for example. And how these groups of cells then may be wired together when there is hyperactivity and lead to, for example, things like interictal spikes. So these kind of thoughts and new research directions combined with the new technologies, I think, will lead to breakthroughs for epilepsy research in general and LGS in particular. Thank you.
Great, thank you, Ivan. And it's really important to keep on reminding us, there's all these new technologies out there, and to apply them to these different models and to really understand LGS and all of epilepsy in a way now that we couldn't have just a few years ago. So I think that's great. So I think we all agree with you on those points. So you're going to kill our discussion because we have nothing to disagree about then. Just